Hello student, welcome to EPG Pathshala. I am Dr. Viswanath Tiwari from Central University of Rajasthan. Today I am going to talk about a module that is mechanism of action of trypsin, ribonucleases and carboxypeptidases. This module has been taken from paper Molecular Enzymology and Protein Engineering. As you know, enzyme in the cell play a very important role in the physiology of the cell. They are important for maintaining the live cell. So the objective of this module is to understand three enzymes out of the thousands of enzymes present in the cell. Those enzymes are trypsin, ribonucleases and carboxypeptidases. So in this module, first we will have a brief introduction about these enzymes. Then we will discuss substrate specificity of trypsin, different isomers or isozymes of the trypsins. Then we will discuss detailed mechanism of trypsin. After discussion of this trypsin enzyme, we will discuss the detailed mechanism of carboxypeptidases, and in the last we will discuss the detailed mechanism of ribonucleases. You know that enzyme converts substrate into the product, and rate of conversion of substrate into the product is slow because of very thermodynamics and kinetic barriers which is involved during conversion of substrate into the product. And transition state is present when substrate is converted into the product. So the transition state is a state which observed during chemical reaction have highest free energy and minimum stability. And a specific form of the transition state depends on the mechanism of a particular reaction. Most of the reactions are slow because the substrate do not have sufficient energy to cross this activation barrier or transition state barrier. An enzyme facilitate the conversion of substrate into the product. So you can define enzyme are the biocatalyst that has the efficiency, a specificity and have a mode of regulation. So in this module we will discuss about efficiency, specificity of trypsin, carboxypeptidases and ribonucleases. Trypsin is a pancreatic serine protease enzyme that hydrolyzes protein with substrate specificity based upon the positively charged lysine and arginine side chain. The enzyme is secreted by the pancreas and take place in the digestion of dietary protein and other biological processes. It has a size of approximately 25 kilodalton and produce in inactive zymogen form, that is trypsinogen. Isoelectric point varies widely in the different form of trypsin. Both cationic and anionic form of enzyme may exist in many organisms. Trypsin and its proenzyme do not undergo any post-translational modification except the proteolytic processing required for its activation. The trypsinogen is activated by removal of a terminal hexapeptide to produce single chain beta trypsin, in which the catalytic residues bridge between two beta barrel domains. The subsequent limited autolysis give other active form having two or more peptide chain bound by disulfide bond. There are two predominant forms, namely alpha trypsin having two peptide pairs and beta trypsin having single chain. The surface loop at amino acid 185 to 193 which influence specificity despite not making direct contact with the substrate. A high affinity calcium binding site is required for stability, and when not present, autolysis occurs. This site is formed by loop glutamic acid 70 to glutamic acid 80. The protein has 6 conserved disulfide bonds at position 15 with 145, 33 with 49, 117 with 218, 124 with 191, 156 with 170, and 181 with 205. The autolysis loop located at amino acid 145 to 151 is very flexible in both trypsin and trypsinogen. Cleavage of this loop at lysine 145 produces the alpha form, which retain the some catalytic activity. The active site residue are also known as catalytic triad of trypsin, consist of histidine 57, spartic acid 102, and serine 193. It functions at optimum pH of 7.5 to 8.5. Out of these 3 catalytic triad residues, the serine 195 act as a nucleophile in the cleavage reaction, producing an acyl enzyme intermediate. Histidine 57 is thought to act as a journal base. Spartic acid 102 is believed to stabilize the correct tautomer of histidine 157 and is provide comprehension for the developing positive charge during catalysis. Replacement of spartic acid to asparagine result in a 10 to the power 4 fold, that means 10,000 fold less efficient enzyme than wild type enzyme in the presence of synthetic substrate with the amino-methylcoumarine leaving group, whereas relocating aspartic acid 102 to 214 yield a protease that possesses 0.5% activity of wild type enzyme. It shows that the spartic acid 102, although it is not involved in direct reaction, but that plays significant role when you're talking about the catalysis of the trypsin. Histidine 57 to alanine mutation or serine 195 mutation exhibit 105 to 106 fold less activity than wild type on the synthetic substrate. The residual activity in these mutant is due to other structural features in the proteases which help to stabilize the tetrahedron intermediate. Trypsin is an endopeptidase which cleaves C-terminal of lysine and arginine amino acid. That means you can say that they cleave a peptide after arginine or lysine. The important thing is, if arginine and lysine is followed by the proline, then the cleavage doesn't take place, because proline donates its imino group in the peptide bond formation, which alter the configuration of the peptide bond between arginine and proline or lysine and proline. Hence, they are not cleaved by trypsin. So, the presence of acidic residues either side of the cleavage site also slows the rate of hydrolysis, because arginine and lysine are the basic residues. So, if they are surrounded by acidic residues, they will interfere in the catalysis of the trypsin. So, the cleavage process is commonly referred as trypsin proteolysis or trypsinization, and the protein that undergoes digestion with trypsin are said to be trypsinized. After discussion of the mechanism of trypsin, let's have discuss different isozymes of the trypsin or isoforms of the trypsin. So, bovine pancreas expresses 2 forms of trypsin, the dominant cationic and minor anionic form. these protein sequence share 72% identity, while their coding regions share 78% identity. Each of these protein is further processed into alternate form. The catalytic trypsin contain a flexible autolysin between residue from glycine 145 to valine 157, and autolysis of the dominant single chain of the beta trypsin at lysine 148 to serine 149 within this loop leads to the formation of alpha trypsin. Further autolysis of at lysine 193 to aspartic acid 194 leads to the formation of psi trypsin. Both the cationic and anionic trypsin protein are expressed as trypsinogen proenzyme with a 15 residue signal protein, that is M12A15, and 8 residue propeptide, that is F19 to K23. The 3-dimensional fold of all known trypsin is highly conserved. In addition, catalytic triad and region flanking the catalytic triad are highly conserved in different forms of the trypsin. Trypsin is a serine proteases. Serine protease means they utilize serine for the catalysis, and the cleavage the peptide bond C-terminal to arginine and lysine. The preference of these basic side chain is evident from the relative catalytic efficiency of this enzyme, which is 10 to the power 5 times greater than natural amino acid. 10 to the power 5 means 1 lakh times greater than natural amino acid. So you can say that they prefer the basic amino acid 1 lakh times more than the other amino acid. The catalytic efficiency is explained from kcat by km. So if the kcat by km of this particular enzyme is determined, it was found to be 1 lakh times higher. It is also known that the trypsin enzyme have preference towards arginine. So they prefer arginine 2 to 10 times more than lysine. In a small substrate, formation of a acyl enzyme intermediate is important. So what happens when the reaction catalyzed by the trypsin start, the substrate will bind to the trypsin, or you can say the serine of the trypsin. It form an acyl enzyme intermediate, and then this acyl enzyme intermediate is cleaved after cleavage of the peptide bond. So the first step is binding of substrate, that is enzyme, binding of substrate to the trypsin, forming acyl enzyme intermediate. Second thing is cleavage of the peptide bond. Third is release of the product. So the formation of acyl enzyme intermediate is a rate limiting step when you are talking about the cleavage of peptides or amide bond by the trypsin, and hydrolysis of this intermediate is a rate limiting step when you are talking about the cleavage of the ester bond, because trypsin can cleave peptide bond, it can cleave the ester bond. Acylation rate with the substrate is a major specificity determinant, and hence the trypsin is much more important or promiscuous with the ester substrate than with peptides. For protein substrate, binding event may be the rate limiting step which lead to the formation of acyl enzyme intermediate. This is the reason why a particular amino acid is required in the protein which is cleaved by this trypsin enzyme, and in this case the basic amino acid like arginine and lysine is preferred for binding to the trypsin and formation of acyl enzyme intermediate. Mechanism of action of trypsin. So mammalian propeptide, usually hexapeptide, trypsinogen, possesses a consensus sequence for cleavage by endopeptidases. Cleavage of propeptide leads to the disruption of hydrogen bond between histidine 40 and aspartic acid 194, which is followed by rotation of aspartic acid 194 so that it can interact with N-terminal at isoleucine 16. These conformational changes result in the formation of oxyanion hole comprising backbone amide of glycine 193 and serine 195 and the binding pocket. The stabilization of new conformation in the active domain is due to the hydrophobic interaction of isoleucine side chain. In other region of the protein, a small change in the position are made during the process of activation. The substrate form a beta antipolar sheet with the protein site. The substrate specificity is determined by aspartic acid 189 side chain, which lies in the bottom of a binding pocket. The high concentration of acetate, it was found that the high concentration of acetate increased catalytic efficiency of the variant enzyme by 300-fold, which demonstrate that negative charge at the base of the trypsin specificity pocket may be bound with some non-covalent bound ligand. The cleavage reaction by the trypsin involves two steps. The first step produces a covalent bond between carbon of the substrate and hydroxyl group of a reactive serine residue of the enzyme. The acyl enzyme intermediate, which is produced, proceed through a negatively charged transition state intermediate where bonds of carbon have tetrahedral geometry instead of planar triangular geometry of the peptide bond. In this step, peptide bond is cleaved. One peptide produced is attached to the enzyme in the acyl enzyme intermediate, and other peptide produced rapidly diffuses away. In the second step, that is deacylation, where acyl enzyme intermediate is hydrolyzed by water molecule to release the second peptide produced with a complete carboxy terminal and to restore the serine hydroxyl of the enzyme which undergoes acylation during the reaction. This step also proceed through a negatively charged tetrahedral transition state intermediate. This step also proceed through a negatively charged tetrahedral transition state intermediate. In this way, the first step is a binding of your substrate to the trypsin producing acyl intermediate. The second step is release of the product and the recovery of the enzyme. After discussion of trypsin, let us have discussed the second enzyme. The second enzyme is carboxypeptidases. So carboxypeptidases are zinc containing enzyme that catalyzes hydrolysis of C terminal to the peptide bond. Carboxypeptidases are secreted as a zymogen by the acinar cell of the pancreases, and zymogen form of this carboxypeptidases are activated by trypsin in the small intestine. Biochemical and X-ray studies have revealed that zinc atom, zinc is a metal, so zinc atom is essential for a catalysis by binding to the carbonyl oxygen of the substrate. As you know that the substrate of carboxypeptidase is protein which have the peptide bond which is having carboxyl group and amino group. So carbonyl oxygen which comes from the carboxyl group is important and it is stabilized by the zinc when carboxypeptidase is going to cleave the protein. So you can say that carboxypeptidase is zinc dependent enzyme. So binding of substrate with the carboxypeptidase weaken the carbonyl bond by abstracting electron from carbon atom and thus facilitating the cleavage of adjacent peptide bond. in a large single domain structure comprises a mixed beta sheet of 8 beta strand with the alpha helix on both sides. Some of the loop regions are very long and curl around the central theme of the structure. The 4 central strands of beta sheets are parallel and have strand order 8, 5, 3, 4. The strand order in the reverse once, and then there is a switch point in the middle of this beta strand between beta strand 5 and 3, where the active site is expected to be located. This is a location where catalytic essential zinc atom is found. Zinc atom is present at this switch point where it is firmly anchored to the protein by 3 side chain ligand, histidine 89, glutamic acid 72 and histidine 196. The last residue of beta strand 3 is the residue 66. So, the 2 zinc ligand histidine 69 and glutamic acid 72 are at the beginning of the loop region that consists this beta strand with its respective alpha helix. The last residue of beta strand 5 is the histidine 196, which binds with third ligand. In the structure of carboxypeptidases, loop region adjacent to the switch point do not provide a binding curvature for the substrate, but instead accommodate active site zinc atom. This zinc atom and the active site are in the predicted position outside the switch point for the 4 central parallel beta strand, even though these beta strands are only a small part of total structure. This sort of the arrangement in which the active site formed from parallel beta strand is flanked by anti-parallel beta strand has been found in a number of other alpha/beta protein with mixed beta sheets. One of the carboxypeptidase, that is carboxypeptidase B, is again a metallocarboxypeptidase that catalyzes the hydrolysis of basic amino acid lysine and arginine and sometimes ornithine from C-terminal position of the polypeptides. In this reaction, you can see that poly-L-lysine is cleaved. Carboxypeptidase B is highly specific for lysine and arginine, but shows greater affinity for arginine. It can also act on valine, leucine, isoleucine, asparagine, glycine and glutamine, but at slower rate. The difference in the specificities between carboxypeptidase A and carboxypeptidase B can be attributed to the residue 205, glycine 241, aspartic acid 253 in carboxypeptidase B. So, you can say that carboxypeptidase B have serine, glycine and aspartic acid residue as compared to glycine 207, isoleucine 243 and isoleucine 255 in carboxypeptidase A. Although the number is different, but if you see the topology, you will find that these residues are present in the same position in the 3-dimensional topology state. So, the major form of the carboxypeptidase B is found in monomeric form with 1 zinc per mole of the protein. The residue coordinating the zinc residues are conserved, including 2 histidine, 1 glutamic acid and 1 water molecule. The active site residues includes glutamic acid 378 and tyrosine 356. After discussion of the second enzyme, carboxypeptidase, let's have discussed the ribonucleases, that is the third enzyme. So, ribonucleases are also known as RNases, are the type of nucleases that cleaves RNA into the smaller fragment. So, ribonucleases are further divided into 2 groups. The first is exoribonucleases and second is endoribonucleases, which comprises several subclasses. Within EC 2.7, if it is phosphorylated enzyme, or in the EC 3.1 if it is a hydrolytic enzyme. Major endoribonucleases includes RNase A, RNase H, RNase 3, RNase L, RNase P, RNase 5M, RNase T1, RNase T2, RNase V, or you can say RNase 5, and RNase U2. The exoribonucleases includes RNase PH, polynucleotide phosphorylase, that is also known as PNase, RNase R, RNase D, RNase T, oligoribonuclease, exoribonuclease 1 and exoribonuclease 2. The endoribonuclease RNase A, or you also say that pancreatic ribonuclease, was the first enzyme and third protein for which a correct amino acid sequence was determined. It is a small protein with molecular weight of 13.7 kilodalton, and mature enzyme have only 124 amino acid with no carbohydrate. The important point of this ribonuclease is they have 19 out of 20 amino acid found in the protein, and the amino acid which is not present in the ribonuclease is tryptophan. The three-dimensional structure of ribonuclease A is fully encoded by its amino acid sequence. All 8 human RNase A like genes are encoded on chromosome 14. Each encodes a secretory signal sequence that contain a invariant catalytic triad of 2 histidine and 1 lysine with a conserved motif of CKXXNTF, where X can be any amino acid. The amino acid sequence of many RNase A homologue has been identified, and from sequence and their distribution over a range of species, it can be established that RNase A is a modern protein that is evolved rapidly. The shape of the ribonuclease protein resembles with the kidney and with the active site residue lying in the cleft. The secondary structure has long 4-strand antiparallel beta sheet and 3 short alpha helices. RNase A is known to have 4 disulfide bonds, which is essential for stability of native enzyme. 2 of these disulfide bonds lies between a alpha helix and beta sheet and contribute more to the thermal stability than other 2. The RNase B is a glycoprotein containing at single oligosaccharide consists of 6 residue of mannose and 2 residue of N-glucosamine at asparagine 34. It is known to catalyze cleavage of phosphodiester bond between 5' ribose of nucleotide and the phosphate group attached to the 3' ribose of an adjacent pyrimidine nucleotides. This cleavage form a 2' 3' cyclic phosphate, which is then hydrolyzed to corresponding 3' nucleoside phosphate. The RNase is found in greatest quantity in ruminant pancreases. The major component of crystalline enzyme is RNA A minor component is RNA B. If you compare RNase A and B, you will find that RNase B is a glycosylated form of RNase A. What is the specificity of this ribonuclease? So this ribonuclease enzyme is specific for pyrimidine nucleotide linkages, and this reaction is take place in 2 steps. In first step, the 3' 5' phosphodiester bond is cleaved while generating a 2' 3' cyclic phosphodiester intermediate. In the second step, a cyclic phosphodiester is hydrolyzed to a 3' monophosphate group. The first step is non-specific with respect to the nitrogenous base of the substrate, but the second step is very specific for pyrimidine nucleotide with terminal 2' 3' cyclic phosphate. the RNase B has the same specificity as RNase A towards both cyclic cytidylate and yeast RNA. RNase A shows a preference for larger substrate. The enzyme cleave at cytidine residue twice as fast as uracil residue. The threonine 45 has been found to be most important for mediating the pyrimidine specificity, both by forming the hydrogen bond with pyrimidine base and strictly excluding purine base. The side chain of aspartic acid 83 is important for stabilizing the transition state during the cleavage of uridine containing substrate. This residue has no effect on the kinetics of cytidine cleavage. Let's summarize what we have discussed in this module. So we have discussed about mechanism of action of trypsin, chymotrypsin and ribonuclease enzyme. We have also discussed what is the specific amino acid required in the substrate of trypsin and carboxypeptidase. We have also discussed about a specific nucleotide requirement of ribonuclease, and we found that the trypsin and endopeptidase cleaves peptides on the C terminal of arginine and lysine, but if the proline is not present at the carboxyl site of the cleavage site. Carboxypeptidase B is a basically type of carboxypeptidase. It catalyzes hydrolysis of basic amino acid such as lysine and arginine towards its C terminal end of the polypeptide. We also seen that ribonuclease catalyzes cleavage of phosphodiester bond between the 5' ribose of a nucleotide and the phosphate group attached to the 3' ribose of a adjacent pyrimidine nucleotide. So in this way, this module explain you about the diverse enzyme kinetics of different enzyme, their specificity, their efficiency. These three enzymes are very important for the survival of human being.